Our next speaker is Dr. Ashley Webb, who's an associate professor at the Buck Institute, and her research is focused on the molecular mechanisms of brain aging and neurodegeneration.
Thank you. All right, great. Thank you so much to the organizers for the opportunity to present today. It's great to be right here at home at the Buck Institute presenting. Just a couple of disclosures, though I'm not actually going to talk about any of this particular work today. All right, so as we just heard, cognitive decline, and in particular loss of learning and memory functions, is a major feature of aging, and yet we currently do not have effective therapeutic strategies to combat this decline in learning and memory that is so well known. And in addition to that, as we just heard, there's also an increase in the prevalence of and the rate of Alzheimer's disease and other dementias with age. And again, there are currently very few, no truly effective treatments, very few treatments at all for these diseases. And so these are diseases that are closely linked to aging, and before the age of about 65, for example, with Alzheimer's disease, pretty much nobody gets these diseases, except for in rare cases where there are genetic disorders associated or genetic mutations associated. And a lot of the work that's been done on cognitive decline has been focused on an area of the brain known as the hippocampus. And that makes a lot of sense. The hippocampus is a critical region of the brain that's important for supporting learning and memory functions. This is what it looks like in a cross-section through a mouse brain, which is the main model that we study here. And although the hippocampus has been very well studied in the context of learning, memory, aging, and emotional regulation, our lab studies this area of the brain too. We actually felt that there was another region of the brain that has been hugely neglected in the context of aging, as well as in neurodegeneration. And so a number of years back, we started thinking about this other area of the brain known as the hypothalamus. And so the hypothalamus has a number of diverse functions that are also critical for healthy aging. And these are just some of the functions of the hypothalamus listed here. So for example, the hypothalamus is critical for appetite and body composition, control of the pituitary glands, or top-down control of the pituitary, as well as these other functions, including, for example, sleep-wake cycles and the body clock. So circadian entrainment actually occurs through our hypothalamus. And so despite the fact that pretty much all of these functions that are listed here are well known to change with age, and when we think about healthy aging and what's annoying about aging, it's many of the changes listed here, right? Yet the hypothalamus is hugely understudied in the aging field, in my view. And not only does the hypothalamus control important functions that change with age, there's also evidence that manipulating certain neurons within the hypothalamus can actually impact lifespan itself. And so these are a few of the studies that have been done, though again, relatively understudied area. That's not good. Relatively understudied area in the aging field, but work from Dongsheng Kai's lab at Einstein has shown that manipulation of NF-kappa B signaling in certain neuron types in the hypothalamus can extend lifespan. Shinamai's lab, studies over here, have shown that another area of the hypothalamus can be modulated via modulation of sirtuin function, and that can extend lifespan. And then a whole body of work from Andre Bartkey's lab studying the growth hormone axis has shown that manipulation of the hypothalamic pituitary axis can extend lifespan in male and female mice as well. So despite the fact that, in my view, there are so many reasons to be studying hypothalamic aging, when we started our work in this area, there was actually very little known about the molecular changes that occur in the hypothalamus. And so this is a problem that a number of individuals in my lab teamed up to tackle. And my lab used to be located at Brown University up until about a year ago. And so some of these individuals started this work at Brown, and then some of them are here at the Buck. But this is a fabulous team that we have working on this problem. All right, a little bit more about the hypothalamus, and to really drive home the issue with the hypothalamus, which I think makes it very interesting to study, but also very complex, is that this is a highly diverse region of the brain. There are a huge number of different neuronal subtypes that are found in this brain, and they have a large number of diverse functions. And so that's basically what I'm trying to drive home here with this diagram, which is representing the different subregions of the hypothalamus. And just to highlight a few subregions that we're particularly interested in my lab, the suprachiasmatic nucleus is the master clock that controls our circadian rhythms and our sleep patterns. In addition, the arcuate nucleus is critical for energy balance, reproduction, growth hormone regulation, and the mammillary body is important for learning and memory functions, and it actually connects up with the hippocampus. And so each of these subregions also includes several neuronal subtypes that are important for each of these functions. So you see what I'm getting at here in terms of how complicated and complex this region of the brain is. And so when we started to get into this work, this was around the time that single-cell RNA sequencing was coming online, and we thought, actually, this is a perfect time to actually get into this diverse area of the brain and to better understand what's going on with the different neuronal subtypes, as well as more globally in the hypothalamus. And so our initial experiment was a very simple experiment where we just did single nucleus RNA sequencing on 3-month-old mice and 24-month-old mice. And I won't get into too many of the quality control details here, but we have a high quality data set that's now published. And the green cells here, these are the neurons that we're particularly interested in this study. All right, so what happens to neurons with age in the hypothalamus? Due to time constraints, I won't have time to talk about any specific neuronal subtype in the hypothalamus, but I'll give you some sort of broad take-home messages here. So in this heat map, every row is representing a different subtype of neuron in the hypothalamus, and every column is representing a molecular process and how it's changing with age. And so the first point that I want to make here is that you can see that some neuronal subtypes are actually very stable with age, and they seem to be more resilient to the aging process, whereas other subtypes of neurons are changing a lot with age, like the ones in the middle here. And so those neuronal subtypes seem to be a little more vulnerable to the aging process. Now, in addition, a second point that I wanted to make here is that when we look at the columns, which remember are the molecular processes that are changed with age, there are some molecular processes that are relatively unique to a certain subtype of neuron, whereas there are other molecular processes that are changed across most of the neuronal subtypes in our analysis. And interestingly, when we look at what types of molecular processes those are, there are processes involved in neurodegeneration as well as changes in metabolism. And so we think that these are the molecular signatures that are actually causing these neurons to become more vulnerable to neurodegeneration during aging. All right, so one thing that I didn't tell you was that when we started this initial experiment, where we just did the simple sequencing of the young and the aged mice, is that we decided to sequence just female mice. And there are a lot of reasons for that I'm happy to talk about afterwards, but it led us to a really intriguing discovery, which was that the most upregulated gene in our neurons with age was a long non-coding RNA known as EXIST. And it wasn't just when we looked at the neurons in aggregate that we saw this change. We actually saw this when we looked across all of our different neuronal subtypes. So EXIST, long non-coding RNA, seemed to be the most upregulated gene in the female hypothalamus with age. Why is this interesting? Well, for us, this was a little bit of a surprise because EXIST is something that I think of as sort of a developmental gene. It's involved in X chromosome inactivation early in life. And so as you know, males are XY, females are XX, and the way that we deal with this situation of having 2 X chromosomes in females is through dosage compensation. And so early on, during embryogenesis, there is a long non-coding RNA known as EXIST that's expressed just from one of the X chromosomes. And that's the X chromosome that's going to be silenced. And so EXIST actually coats the X chromosome that will be silenced and induces an epigenetic cascade that actually causes that chromosome to be transcriptionally silenced throughout life, and it's tucked away into the side of the nucleus along the nuclear periphery, and it's supposed to stay silenced, again, throughout the lifetime. And so it was a surprise to us that this EXIST RNA was actually the most altered with age in our study. And so currently we're working on understanding the mechanisms underlying the changes that we see. But in the meantime, as we're doing those mechanistic experiments, we wanted to ask whether there was something special about the X chromosome itself in females, and something about EXIST itself that might help us use it, for example, to predict age of a cell or to be used as a biomarker of aging. So we turned to a machine learning approach, and we took just the genes on the X chromosome as input in our machine learning, and we then inputted these genes into a number of supervised machine learning algorithms, and we simply asked, can we predict the age of a cell as young or aged using just X chromosome features? And when we did that, it turns out it actually worked pretty well. And in particular, it worked when we used the XGBoost algorithm shown here, and what we found was that about 85% of the time, we could correctly we could correctly call an aged cell as aged or predict that an aged cell is aged. And when we looked under the hood, it turned out that the number one feature on the X chromosome that could allow us to predict the age of a cell was actually the exist gene itself. All right, so we have a lot of work to do. This was a pretty unexpected discovery on our end, but these are a few of the questions that we're now tackling in the lab. So first of all, we want to know now, are there sex-specific transcriptional and epigenetic changes during aging in the brain? So we were looking at females before. We do care about males as well, and so now we're incorporating males in our analysis as well. As I mentioned, we're also interested in understanding the mechanisms underlying exist changes with age, whether it's driving, aging, or simply a compensatory response. And then finally, we're very interested more broadly in looking at what are the cell type specific features in the aging hypothalamus. So can we actually use machine learning to get into different cell types of the hypothalamus and actually discover how these different cell types are aging, since we have these single cell data sets. So I'll just talk a little bit about the 2 points here in red. And so we've gone back and done all of the sequencing again, both in males and in females, and we also have the epigenetic state of the chromosome in males and females as well. And I'll just give you one take-home message from this data set. The take-home message is that when we looked at glia and we compared differential expression in males and females, we actually saw a lot of similarities between the sexes. And what gave us a surprise when we looked at neurons, and this includes both excitatory neurons as well as inhibitory neurons, we actually saw pretty much a lack of correlation in terms of how the neurons were changing with age in males and in females. And so these data are unpublished, and we're digging deeper into the data set to try to understand what's going on here. But we think there's going to be a lot of new insights, we hope, in terms of understanding sex-specific neuronal aging. All right, but in the last few minutes, I want to tell you about the new machine learning pipeline that we have generated to look at cell type specific aging in the brain and specifically in the hypothalamus. And this is a collaboration with Ritambhar Singh, who is a fabulous computer scientist at Brown University, as well as my graduate student, Dodo Yu, who's now here at the Buck. All right, so we simply wanted to ask, can we learn from our data set what classifies a cell type as aged or old? And then can we use that classification, that classifier, to then discover the gene expression networks that are underlying aging in different cell types using single cell data sets? So again, our input here is actually, it's similar to what I showed you before, but instead of using X chromosome genes, we're actually using the highly variable genes in the data set. And these genes are important because they're the actual genes that separate the clusters on the UMAPs that we use. And so these highly variable genes are important because they actually define the different cell types in the data set. And we want to get at cell type specific signatures here. So we took those cell by gene matrices, fed them into a number of different machine learning methods, as well as a neural network analysis, and we performed a classification just in terms of young or old. And the nice thing is that we can then go and do model interpretation, and that's what actually allows us to discover the gene expression networks for these different cell types. Now, when we initially did this, we were actually very disappointed. The performance of our models, it wasn't very good at all. And so we really had to take a step back and think about, like, what exactly are we doing here with this transcriptomic clock? And not only that, we had to think about the fact that in general, as we heard from David Furman earlier, transcriptomic clocks don't work nearly as well, for example, as methylation clocks or the epigenetic clocks that are out there. And so we stepped back and took a look at our cell by gene matrix, and we thought about the fact that in terms of transcriptomic data, really what we're dealing with here is continuous data. And you can see that illustrated here. And then we thought, you know what, when we look at the methylation clocks that are published, those aren't continuous data. For the most part, those are binary data, right? DNA methylation is binary. So we drew some inspiration from the clocks that were already published, and we decided, you know what if we just binarize our matrix? And when we do that, this is what it looks like. And I think right away you can see what it's doing to the data set is basically denoising the data set. So we then went back and we took this binarized data set and we ran it through the same pipeline, and to our surprise, our performance shot up to about 95%, and this was across different machine learning models. This is elastic net performance shown here. And so we were very happy with this performance, and we call this new method cell by age. All right, so what do we do in the cell by age now? So now we're trying to go into the specific cell types in the hypothalamus and discover how they're aging. We have run the model across the major cell types of the hypothalamus, and we found that we can predict age across major cell types. And we compare the genes that are actually driving the model in these different cell types. What we find is we have cell type specific gene expression changes. And this is what we were going for. So in this analysis, we only found about 4 genes that were changed or driving the model across all of the different cell types, whereas most of the underlying coefficients were actually cell type specific, for example, shown here. All right, so this is cell by age, which is a transcriptomics-based machine learning method, and it's used to predict single-cell age using single-cell RNA-seq data sets, and it's useful because it can discover cell-type-specific aging signatures, as well as evaluate interventions. So this is something that we've done more recently. We took a data set from Anne Brunet's lab at Stanford, where she had exercised mice, and we asked whether cell by age could predict rejuvenation of cell types in the brain, and in fact, we found that it could, and this is published. All right, so why is this important? Well, I think it's very important that we establish these single-cell methods or clocks because most of what we're looking at in terms of pathways that modulate aging tend to be pathways that are expressed across all different cell types, right? So when we think about, the major regulators of aging, insulin, IGF, FOXO signaling, TOR signaling, those pathways are relatively ubiquitous. Yet we know that distinct cell types across the body aren't aging in the same way. So we need to understand the changes in those more specific cell types to understand how they integrate with these age-associated pathways in order to lead to disease. All right, so that was the second part of the talk, the cell by age, and in the first part of the talk, I told you about the work that we're doing in the mouse hypothalamus with age, and then we have discovered transcriptional and epigenetic changes in the male and female hypothalamus, and specifically sex-specific differences. We've also found elevated expression of EXIST in the female brain, and we believe this may be a new biomarker for single-nucleus transcriptome, a biomarker for aging in single-nucleus transcriptome data sets. And we are currently working on the mechanisms involved. All right, and I already acknowledged the people that did the work today, and I will just thank my sources of funding, as well as, again, the organizers for the great meeting at the Buck. Thanks. And I can take questions.
Very nice talk. I have a question about kind of a generalization of this idea of the release of EXIST. Do you know any data that suggests that this could happen in other part of the brain first, but even other organs where maybe cells live very long as well?
Yeah, so this is a great question. So is the EXIST change happening in other cell types? So we see it in the hippocampus as well, in the neurons specifically, and we see it's mostly enriched if you do single nucleus sequencing because that's where most of the EXIST is. And I have had some conversations with others who have seen it in the context of the immune system, though my sense is that the immune system is a little bit different given that the turnover, whereas with the neurons, one feature of the neurons is that they're early born in life, and then what we're seeing is probably more like an epigenetic drift with age, whereas in the immune cells, those cell types have the opportunity to reset when they divide. So I suspect that the immune system might be a little bit different, but we've seen it in other cell types in the brain as well, and we've actually seen it in some human data sets in Alzheimer's disease.
Yep. Great talk. So I have a similar question, a related question, I guess. So can you speculate a little bit about what EXIST elevated expression is doing, and is it uncovering expression of a bunch of genes, and what do those genes do, and what does that mean for aging?
Yeah, what does this all mean? Yeah. So I can speculate. So what we see in our most recent data set, where we've looked at both the epigenetic state as well as the RNA levels across the X chromosome, is that there's a general upregulation of expression on the X in females, not in males. And there also seems to be a slight increase in accessibility of the chromatin across the X chromosome in females, but not males with age. And so I actually suspect that exist may be a compensatory mechanism, so the upregulation may be a compensatory mechanism due to a loss of heterochromatin that occurs with age, and that's been seen in the context of senescence as well, this deheterochromatinization that can occur. And so again, I don't have the functional data to back that up, but that's certainly what we see that in our data sets, and it correlates with aging, but I don't have the killer experiment yet, so stay tuned.
And do you think it has anything to do with the increased prevalence in women of Alzheimer's?
I think it might. I don't have evidence for that either, but I think that there's a link there. I mean, the X chromosome also has an abundance of, or an enrichment of genes involved in neuronal function over autosomes. And so, I suspect that, again, no evidence, but I suspect that there's a link there. And that's the reason, why neurodevelopmental disorders are more prevalent in males, because there are so many genes involved in things like synaptic function that are on the X chromosome. And so males, where they only have one X chromosome, tended to have more neurodevelopmental disorders. So there is this enrichment on the X, and I really suspect that there's something going on there, in terms of neurodegeneration, as well as just normal brain aging.
Sorry, this is potentially a naive question, but I'm just really curious. Just because for this exists RNA, this is speculative because, of course, you don't have functional data yet. However, what if you would just introduce the original exist RNA into the human cells again? What do you think could happen or would happen? I know you're looking at this as a marker of aging, but is this a potential target for therapeutics or it itself?
So far, I think we've shown that it is a biomarker of aging in these single nucleus data sets, at least in the brain and the neurons. I think there is potential for it to be a therapeutic target, but again, we need to understand whether it's driving aging or just sort of along for the ride. So I don't yet know if it's, if it's sort of good or bad, air quotes, if you want to use those, oversimplify it in that way. But this is the question. Yeah, I think it's super important. Thanks.
Hello, thanks for the very interesting talk. I was wondering if you have some data about endothelial cells and neurovascular unit, or just a technical problem because they just are low in number, is difficult technically to obtain, or you didn't find the same results on endothelial cells?
Yeah. So we haven't done much analysis of the endothelial cells. We certainly have them in our data set, and I would actually be happy if anyone is particularly interested, we'd be happy to share the data. Most of it's publicly available, and anything that isn't, I'd be happy to work with people on that, because that's not our main area, but we certainly have the cells in the data set, so yes. But I can't speak to exactly what's going on there. We do see changes, but we haven't looked at that more specifically. All right, thanks.